Hello everyone, today I will be showing you how to perform a homology modeling simulation using InteractiveROSETTA. Homology modeling requires use of the external C++ Rosetta server, so please refer to the tutorial on setting up that server if you have not done so already. This used to be a quite difficult Rosetta submission to set up, so I hope that the graphical tools in InteractiveROSETTA help make this process easier for you. First off, I am going to load two PDB files. The top one, 1ADW, is going to be our template. The structure we are trying to predict will be the lower structure, 1BAW. Now we can select the structure prediction module from the protocols menu. There are not too many controls on this panel, which is surprising given the complexity of running homology modeling at the command line. The first thing we need is the sequence of the structure we will be predicting. Let me show you a neat little trick. It turns out that you can select sequence down here and do a Ctrl+C copy to get the selected sequence copied to the clipboard as sequence data. Now I can paste it over here. Pretty neat, right? Now I have to specify which templates will be used as the scaffolds. There is only one loaded right now and it is the only one I am interested in. If you had multiple templates as scaffolds, you can specify them here. The order of templates indicates the priority. The one at the top is given the most priority and the one at the bottom is given the lowest priority. You can change the order by clicking on one of the templates in the grid and using the arrow buttons to move them up and down. You will notice that when I click on one of these templates, I am given an alignment between this template sequence and the sequence whose structure I am predicting. This alignment is initially generating by muscle, but you will need to edit this yourself. Why? Because the gaps which represent insertions and deletions may have ended up in the middle of secondary structure elements. Obviously you need to move those gaps to loop regions. Keeping track of all of that is tricky, so the structure in PyMol has been recolored to help you out. The purple regions are regions where there is an alignment between the two sequences, so the given sequence will be threaded onto the purple regions of the displayed structure. If you see blue regions, those are the areas that are unaligned in the target sequence, so the structure will be deleted in the final model. Yellow point indicates unaligned regions in the template, where you will be inserting loops for the extra target sequence residues. I want to move these yellow and blue regions out of the alpha helices and beta sheets and into loop regions if possible. Surprisingly, everything looks mostly okay right now, except for a deletion in the first beta sheet over here. I want to move the deletion out here. It is a 2 residue deletion that is right up here. You can click in this window and edit the alignment. The arrow keys are functional to allow you to move around, and if you watch the PyMol and sequence windows you can see that it updates my selection, to show me where I am in the structure. You can use the delete or backspace keys to delete gaps. Then I will move down here and use the space bar to insert the 2 residue gap out here, to keep the rest of the alignment intact. Some alignments can take a while to put together, so there are save and load buttons up here to prevent you from having to generate these alignments over and over again. Finally, I need to specify how many models I want to be returned, and then send it to the server. If you are familiar with Rosetta, you will likely be wondering where the fragment files are coming from. The server takes care of all of that for you. So you do not need to go to Rosetta to get fragment files. This process takes about 30 minutes, so I will see you again when this process is done. Hello again. Looks like my job is done, so I will download it to my computer. Here is the first model generated by Rosetta. It is not perfect, but this was just a dummy example that I set up quickly to show you how to use homology modeling. In a real application I would obviously choose my templates more carefully. Anyway, that is how you run homology modeling in InteractiveROSETTA. See you again in the next video.